Right, I am now recording. Because this is a recording and nobody is with me, I'm going to try to do this very, very fastly to kind of catch up, to make up for a little bit of lost time. Because since it's a video, obviously you have the ability to pause it and restart it, um, and even slow it down if you want to. So here we go. Let's jump into it. Before I do jump into it, I just want to let you know I am working from home today. Obviously, the campus was on a 2 hour delay. Also, my children did not go to school. They were snowed out. So, if you hear any loud noises in the background, I apologize in advance. That is what the reasoning behind that. Anyway, let's jump into this. Let's talk about announcements, reminders. I want to remind you you can now retake exam one. You can take it as many times as you want. It's open book. It's not timed. Yeah, and that's it. Just email me when you're done, once you're satisfied with your new grade, and then I'll go in and average your first grade with your highest grade. So again, there's nothing to lose by retaking the exam. If anything, even if you somehow end up doing worse, we'll just keep your original grade. But again, it's good practice to help you with the prepare for the final. Also, lab. I want to remind you about lab this week. It's an important one. This is the one where we write the lab report. So in a sense, it's almost like two labs, right? Because the lab report is also worth 20 points. So it's very important that you're there. Make sure you're on time. I will be locking the doors at 9 a.m., right? So nobody can come in. If you're late, you're just missing the lab. It's very important that you be there and be there on time. Also, obviously, if you haven't done the pre-lab yet, you're going to want to go ahead and do that. Yeah, that's it for that. Participation points. So do this as we always do. I'm going to say a series of keywords or attendance words or phrases throughout this lecture. You're going to write those down, take a screenshot every time I say one of those, and then send me an email that includes all the attendance words, all the screenshots, and one question about the material that I cover in this lecture. So one is going to be the first word for attendance. Let's move on here. So this is where we left off. We were talking about the really big picture here when it comes to cellular respiration, right? When we're looking at the big picture, for the most part, it's all about stripping away these high energy electrons, right? That's what the first two phases or the first two stages are for, glycolysis and the citric acid cycle, right? We're stripping away those high energy electrons. Those high energy electrons are then carried by NADH and, of course, FADH2, which we talked about previously. But anyway, these high energy electrons are taken to the electron transport chain where they kind of go from protein to protein. And as they go from protein to protein, they're releasing some of that energy. And that energy is being used to make ATP. And as we move forward in the slideshow, you'll see, I'll explain how that happens. But eventually, ultimately, what happens is, like I said on Friday, electrons have to come from somewhere. They have to go somewhere, just like in any kind of electrical circuit, right? And in this case, where do the electrons come from? They come from food. Of course, when you get a test question on that, it's going to be, the answer will be glucose, right? The electrons come from glucose. Where do they go? Well, they are accepted by this oxygen, and the oxygen then, when that happens, that forms a molecule of water. So let's talk about it. Again, like I said up here in this is a basic picture. Whoops, excuse me. These electrons are going from protein to protein. But now let's talk a little bit more about that. These proteins are embedded in the inner membrane of the mitochondria. And this is going to make more sense when I show you a picture moving forward. But yeah, and what you'll be able to see in this picture is you'll be able to see that the membranes are highly folded. This is important. And again, this theme of structure and function, right? So it's highly folded. And the reason for that is because that gives it a larger surface area. So the more surface area it has, the more proteins it can have embedded within the membrane. And obviously, if these proteins are the things that are making ATP, then the more proteins you have, the more ATP you're producing. So here's a picture of what I'm talking about, right? On the left is a drawing of the mitochondria. You can see they've kind of cut into the mitochondria, so to speak. So you can see there's the outer membrane of the mitochondria over here. You can see the inner membrane of the mitochondria. And even though you can't see it in this picture, just imagine, and I'm trying to draw, and that looks horrible, but imagine these proteins that are embedded in the inner membrane. So we already learned about all kinds of proteins already in this course, including transport proteins. Remember, transport proteins allow things to go back and forth, from the inside to outside of something with a membrane. Now, granted, previously we only talked about cells, But as far as this conversation is concerned, this mitochondria might as well be a cell because we're talking about the membrane. Anyway, these proteins allow things to come in and out of the membrane, and that, excuse me, in and out of the inside of the mitochondria, specifically hydrogen protons. And we'll talk about that here in a few minutes when we get to that side. But what's happening again, and even though you can't see it in this picture, these hydrogen ions are going to get pumped against their concentration gradient, right? So they're going to go here to where they'll be trapped between the inner membrane and the outer membrane. So as you already know from when we had discussions about diffusion, for example, right? We have all these hydrogen ions now trapped in the inner membrane, in between the inner membrane and the outer membrane. So what they're going to want to do, and again, this will make more sense later when I show you pictures of it, but then those hydrogen ions are going to want to naturally diffuse back down the concentration gradient and go back to where they came from, which is the inside of the mitochondria. And again, that'll make more sense here in a few slides when I show you a different picture of it. But yes, that is a drawing of the mitochondria on the left. There on the right, you can see the microscope image of a mitochondria, but you can see how folded it is, right? Again, more surface area, more proteins, more ATP. So again, like I keep saying, these proteins, these basically transport proteins, and they are using the energy from these high energy electrons to pump hydrogen ions against their concentration gradient. And in a sense, this is very much just like water behind a dam. Of course, when it's water behind a dam, it's usually a river, and we're not using pumping water against the concentration gradient. But you have all this water behind the dam, you have all that potential energy, and the way we make electricity, right? When we're talking about hydroelectricity, is we just let nature do its thing, right? We let gravity take over, and the water wants to go downstream, right? It just naturally does that. And when we allow it to go downstream, we have turbines, right? And those turbines get turned by the water, and that's how we make electricity. And in a sense, the mitochondria is very much like that. So what we're doing, again, we're pumping these hydrogen ions against their concentration gradient so that they're stuck here in between. You can see the outer membrane here, right? And the inner membrane here. They're stuck in there. And they want to go back down the concentration gradient. And they do. And there's transport proteins. Again, we've already talked about the transport proteins that pump them against their against the concentration gradient, which is how they get stuck in there to begin with. But then there's a different protein that allows them to come back down the concentration gradient. And again, this will make more sense when I show you a picture on the upcoming slide. Now, that protein that I'm talking about, notice I haven't named any proteins in this entire chapter. We've talked about proteins, and we talked about enzymes that make ATP in glycolysis, right? But I didn't give you a name of it. I just told you that we have proteins that pump hydrogen ions against their concentration gradient. Notice I didn't give them any names, right? I didn't tell you what it was named. But this one is important, and this one we're giving it a name. This protein, this enzyme, is called ATP synthase. So when I was using the dam as an example, right? We said the water goes down, it goes downhill, so to speak, downriver, and that turns the turbine, and that turbine is how we make electricity. Well, the ATP synthase is very much like that. Again, in that it allows these hydrogen ions to diffuse back down their concentration gradient. But when they do, it actually does turn, right? There's a little rotating piece of this of the enzyme, and that's where we get our ATP from. Now, for the exam, I want you to be very clear here. You do need to know the name of this enzyme, right? There's not many enzymes where I'm going to ask you the name of them. So if you're taking notes, write that down, right? This is one of those enzymes where you need to know the name of it. All right, let's move forward. This is a picture from the old textbook. I think it's a great picture. It really illustrates what I've been trying to say. What we're looking at here, you can see on the right, we can see this mitochondria, right? Where they've cut into the mitochondria, and we're looking at it that way. And then you can see this little square right here. So we're zoomed in. So what we're looking at here's the inner membrane, which has these, I'll just write an I here. The inner membrane is here, right? And what you can't see, somewhere outside of the picture, would be the outer membrane right here. But these hydrogen ions, again, they're getting pumped against their concentration gradient. So they're stuck in between the outer membrane and the inner membrane. And the energy for that, because remember, when you're going against a concentration gradient, that takes energy. But that's where these high energy electrons are coming from. This is how they're being put to use. All these high energy electrons that were stripped away and harvested during glycolysis and during the citric acid cycle, those are taken here, and they go from protein to protein, as you can see in these pictures. They go from here to here to here. And as they're doing that, they're releasing their energy. And that energy is used, as you can see in this illustration, to push hydrogen ions against the concentration gradient, right? That takes energy. And then again, like I've already mentioned a few times, after that, nature just does what it does. And these hydrogen ions, which are highly concentrated in that area because we keep pumping them up there, they just diffuse right back down the concentration gradient, back into the inside of the mitochondria. And as it does that, like I said, it goes through this enzyme called ATP synthase. And that is what makes the majority of the ATP in the whole entire process of cellular respiration. And again, I'll remind you, because it's the same thing, but a different picture. Electrons have to come from somewhere. They have to go somewhere. Where do they come from? They come from food, technically, but as far as the exam is concerned, we just simplify it and just say it comes from glucose, right? The electrons come from glucose. They are then carried by these electron carriers like NADH and FADH2. And again, as far as you guys are concerned, there's the same thing. They're doing the same job. They're just carrying electrons. But again, electrons have to go somewhere. Where do they go? They are accepted by the oxygen. And when they are accepted by the oxygen, that forms water, a molecule of water. This is what this little scroll and crossbones is for. It just indicates, like, when people die from a lack of oxygen, this is the reason why. This is how oxygen is used, and this is why you die from a lack of oxygen. And that's it. If anybody was on the meeting, I would ask if you have any questions, but there is no one in the meeting, so let's just move forward. Here's another number I want you to know, and I mentioned this in a previous lecture. Cellular respiration generates about 32 ATP molecules per molecule of glucose. Now, the number varies, also varies depending on the source that you're studying. So just know, for the exam, I want you to know the number 32. And as I mentioned in the previous lecture, when you get this question on the exam, this multiple choice exam, the options are going to be at least 10 apart. Meaning, if I don't want you to get concerned like, oh, was it 32 or was it 38?